let's continue along. So here I have a collection of water. I have a glass of water, a bunch of water molecules in here. Water molecules are liquid. They're all holding themselves together due to this hydrogen bonding. And then what I'm going to do is I'm going to introduce some salt, some sodium chloride into this water here. And what do you suppose is going to happen when that sodium chloride goes into the water? Well, what kind of charge does sodium have on it? Positive. So where do you think that sodium is going to be attracted? Yeah, it's going to be attracted to the negative pole of the water molecules, right? So I put the sodium chloride into the water, the sodium is going to be attracted to the, the positively, right, because it's an ion, and the chloride is going to be attracted to the chloride is going to be attracted to the positive part, the hydrogen part, and what's going to happen is that's going to pull the sodium chloride molecule apart. So when I put that salt into the water, it gets pulled apart, and that's what we call dissociation. It gets dissociated, okay? And when something gets dissociated, or when something is attracted to the polarity, the polar nature of the water, we say that the water dissolves it. It got dissolved. Okay? And so, ions and other polar molecules are easily dissolved in water. Water can easily dissolve ions and polar molecules. And it is because water itself is a polar molecule. Does that make sense? Water is polar, therefore it can dissolve polar or ionic things. And that is why we say that water in biology is the universal solvent. And a solvent is something that dissolves stuff. And if something gets dissolved in a solvent, it is called a solute. So the sodium chloride would be the solute. All right? So the solute gets dissolved and the solvent does the dissolving. Now, does everything dissolve in water? Can water dissolve everything, even though we call it a quote-unquote universal solvent? No, non-polar molecules. Molecules that are not ionic, are not very ionic, don't dissolve in water. And this is in fact why you guys have put like an oil, like have you ever like put some olive oil in water? Do they mix up? Does the water dissolve that oil very easily? No. Why? Because fats are non-polar. And non-polar molecules will not dissolve in water. Does that make sense? That's why water does not dissolve oils. But it can dissolve other things like table salt. Okay? So for us to digest fats and oils, which are fats, lipids, we have to break them down, we have to modify them, we have to make them into something that's easier to dissolve or easier to be transported in the digestion process. You guys okay with that? All right, make sense? Okay, cool. So now what I want to do is I want to talk about a special case of this dissolving. And the special case of this dissolving is anything that, when it becomes dissolved in water, okay, anything that, and let me just give you an example. Let's say that I have hydrogen, that's really hard to see. I have hydrogen chloride, HCl. Okay, so the H is going to be positive, right? It gave its electron up to the chlorine's negative. You guys cool with that? I put this hydrogen chloride into the water. What do you suppose is going to happen? The hydrogen that's positively charged is going to be attracted to the... All right, so the hydrogen is going to be attracted to the positively charged oxygen. And the chloride that's negatively charged is going to be attracted to the hydrogen on the water. Does that make sense? And it will dissociate, right? whenever a molecule dissociates and releases or donates, okay, anytime a molecule donates a hydrogen ion, anything that donates a hydrogen ion when it gets dissolved in water is what we call an acid. An acid, for our purposes, okay, this is known as the Bronsted-Lowry definition. There are other definitions, but for now this will work. Any substance that donates hydrogen ions or protons. What is the difference between a proton and a hydrogen ion? Nothing, right? A hydrogen ion is a proton, right? Because what is hydrogen? Hydrogen is a proton and an electron, right? And when it loses an electron, you're just left with a proton. So that's equivalent, same. Okay? Hydrogen ions and protons are the same thing. That's what an acid is. You guys okay with that? Okay, so when your body makes some substance that donates hydrogen ions, okay, it breaks down and it releases hydrogen ions. That's what an acid is. So what do you suppose a base is? A base is something that accepts or takes hydrogen ions. So bases accept hydrogen ions or protons. Okay? So something like a hydroxide, OH negative. An OH negative could accept an H positive, and that would make what? H2O. Yeah, okay, so hydroxide would be a base. Hydrogen ion would be the acid, okay? I could have bicarbonate, HCO3 negative. It could accept a hydrogen ion as well, and it would make H2CO3, okay? Are you guys okay with what an acid is and what a base is? So something that is acidic donates hydrogen ions or donates protons. Something that is basic accepts hydrogen ions or accepts protons. You guys okay with that? Now, because this is a very important concept in biology, okay? Acidity, because when things go wrong, acids can be produced, and we'll talk about that in more detail as we get further on in the course. So we have to measure acid. Okay, it's very important that we measure the level of acid, okay, in the body. And the way we do that is through something called the pH scale. Little p, big H. All right. And to understand the pH, I'm going to go back to the periodic table of elements. Okay? The periodic table of elements, if we look at hydrogen, do you see a decimal number anywhere? Probably on top of the hydrogen? It's 1.0.
107.
That is the atomic mass. Okay? And what we have found out is, if I have a very large number of that, okay, a huge number, a massive number, and you guys remember we talked about this scientific notation in lab, and it's 6.02 times 10 to the 23 atoms of hydrogen. So I have to move this decimal point over 23 times, whatever that number is. That massive number is what we call Avogadro's number, or the common statement is the mole. A mole is 6.02 times 10 to the 23. If I had a mole of hydrogen atoms or hydrogen ions, I would have 1 gram of hydrogen. Okay. If I have a mole of helium, okay, helium is what, 4 point something?
Yeah.
Is that right? 4? 4.0 something, right? If I had a mole of helium, I'd have about 4 grams of helium, okay? And so on and so forth, okay? You guys, so far so good? So what I'm going to do is,
Chris,
yes, sir.
1 mole is 1 gram?
No, just of hydrogen.
Okay.
But if I had a mole of hydrogen, I'd have about 1 gram of hydrogen. If I had a mole of helium, I'd have about 4 grams.
So if you had a mole of sodium,
It would be
Oh,
whatever 22, about 22. Mole of carbon would be about 12 and some change grams. Okay. So if I have if I have an Avogadro's number, a mole of an atom, I would have its atomic mass in grams, more or less, is the conversion.
is it the name for a unit of AMU?
What do you?
A mole?
No, it's a number. Mole is 6.02 times 10 to the 23 atoms. So if I have 6.02 times 10 to 23 hydrogen atoms, I have about 1 gram. If I had this 6.02 times 10 to 23 helium, I'd have about 4 grams of helium, and so on and so forth. Yeah. So there we go. That's the mole. Okay. So if I had a liter, so let's go to this water. Imagine that I have a liter of water in here. Okay. I could put a mole of hydrogen ions into that liter. Okay. And that gives me an acid concentration of 1 mole per liter. That would be massively, that would just be so acidic you could not even imagine how acidic that would be. And we just don't run into that type of acid level in nature. What we do run into is we run into fractions of a mole, okay? We run into like 10 to the negative 7 moles per liter of hydrogen ions, okay? That would be a more reasonable situation to have. So if you go into the hospital and someone were to come up to you and say, hey, your patient has 10 to the negative 7 moles, so 10 to the negative 7 of 6.02 times 10 to the 23 of hydrogen ions per liter in their body, does that mean anything to you? Does that make any sense? What do you guys think? No, that's a tiny number, right? That's just, it doesn't make any sense. So we need some way of simplifying these crazy numbers, okay? And one way we can do that is we can take, are you guys familiar with the concept known as a logarithm? You guys heard of that, a logarithm? Specifically the log base 10, okay? So for example, what's the log base 10 of 10? What's the log of 10? What's that?
1.
Someone said it, 1. Yeah, the log base 10 of 10 would be 1, right? Because how many 10s do I need to get to 10? 110, right? What would be the log base 10 of 100? That would be,
10 times.
no, 10 times 10 is, so it would be 2, right? I need 2 10s, right? 10 times 10, right? What would be the log base 10 of 1,000? 3, right? 10 times 10 times 10. Are you guys seeing the trend here? What would be the log base 10 of 10,000? 4. 100,000. 1,000,000. Right? This was just an extension of the metric system that we talked about earlier, right? Remember we talked about the metric system? Yeah. So what I do is, whatever my concentration of hydrogen ions per liter is, okay? So if it's 10, say 10 to the negative 7 moles per liter, okay? I take the negative logarithm base 10, not the log base 10, but the negative, okay? The negative log base 10. And what that allows me to do is that allows me to get rid of the minus, first of all, and then just one number pops out. 1, 2, 3, 4, 5, 6, 6.2, whatever. Just a simple number pops out. Okay, let's see, the negative logarithm of 10 to the minus 7 would be 7, I believe. Okay. That number there is what's called the pH. Okay, so the pH scale, when we measure somebody's pH, pH is the negative log base 10 of the hydrogen ion concentration in the human body. And the reason that we do that is because we're talking about numbers that are so big and so small that we do this logarithm. We take the log of it to make more sense of it. Does that make sense? Sort of? All right. Because it's easier to go, oh, my patient has a pH of 7.4. Okay, that makes more sense. So now, what we do is we get our pH scale. Erase all this stuff here. And our pH scale typically goes from 14 to 0. All right. So 0 and 14, right? So all the way down here, way down here at 0 would be what? More or less acidic? Negative logarithm, though. More, right? We're taking the negative log, so things flip, okay? So this would, 0, right? 0 would be like 1 mole per liter, right? That'd be really concentrated. And then 14 would be like negative 14 moles per liter, something like that. It'd be really not very acidic, okay? And then 7 is about right in the middle. And 7 is what we call neutral pH. Neutral pH, that's neutral. And everything lower than 7 is going to be more acidic. And anything higher than 7 is going to be more basic or alkaline. All right, basic or alkaline. And because this scale is a negative log, it's a base 10, every jump is going to be 10 times more acidic or 10 times more basic. So if I went from a pH of 7 to pH of 6, 6 is going to be 10 times more acidic. Does that make sense? And if I went from a pH of 7 to a pH of 5, 5 would be how many times more acidic than 7? 20. 100, right? 10, 100, 1,000, 10,000, 100,000, million, etc., right? So a pH of 4 would be how many times more acidic than 7? 10, 100, 1,000. A pH of 3 would be how many times more acidic than 7? Right? Yeah, so 10, 100, 1,000, 10,000. Does that make sense? Now a pH of 8 would be how many times more basic than 7? 10 times. 8 would be 10 times more basic than 7. A pH of 9 would be how many times more acidic than 7? 100. A pH of 10 would be how many times more basic than 7? 1,000. Okay. A pH of 9 would be how many times more basic than 6? 1,000, right? 10, 100, 1,000. You guys okay with that? Make sense? Cool. All right, so that's how the pH scale works. It's a lot like the Richter scale for earthquakes, right? It is, okay, 10 times. It's what we call an order of magnitude. So 1 order of magnitude would be 10 times. 2 orders of magnitude would be 100. 3 orders of magnitude would be 1,000, and so on and so forth. Does that make sense? Now, what we need to know is, at what pH does the human body optimally work? Is there an optimal pH for the human body?
Yes.
Yes, there is an optimal pH. And it is slightly alkalotic. Okay, and the optimal pH for the human body is a narrow little range right in here. And it is 7.35 to 7.45. With 7.4 being perfectly normal. Okay? So the human body, for all the chemical reactions to occur normally in the human body, the pH needs to be between 7.35 and 7.45. If the pH is more acidic all of those extra hydrogen ions are going to interact with the chemical systems. They're going to make chemical reactions occur that don't need to occur or inhibit other chemical reactions. Likewise, if I get too basic, okay, if my pH gets higher, and actually I should probably do this to limit confusion, huh? There we go. Since that's acid, that's basic. Likewise, the same is true with being basic as well. You guys okay with that? Does that make sense? Does that make sense what an acid is? Yeah.
So if we have a diet that's very high in acid, will that really affect, like if we drink a lot of coffee or wine or something?
It could.
Or is it fairly?
Well, you'd have to really go crazy because, and I'm not going to be able to talk about this today, but your body has what are called buffer systems. And I believe in Anatomy and Physiology 2, you guys will talk about the respiratory system and the renal system, which are two of the three, and the blood buffer system, the major buffers. So you have buffers that can neutralize acids and get rid of them. But if you're really sick and those buffer systems happen to break down, then you could be in a world of hurt. Or if you do something, like if I stick a tube down somebody's stomach, and I start suctioning, what do you have a lot of in your stomach?
Acid.
Just have a lot of acid. If you vomit excessively or I suction too much acid out of your stomach, I could make you alkalotic. Likewise, if you have too much diarrhea, okay, you actually lose a lot of base. And I just remember you lose base out of your butt and acid out of your mouth. And so someone who has a lot of diarrhea could actually become acidotic because they poop off a lot of that base. So there's definitely some good clinical implications to this stuff. You guys okay with that? That makes sense? Okay, cool. That's the basis of acid-base, and you guys, of course, will talk about it in a little more detail. And again, the reason that this is so important is if you become too alkalotic or too acidotic, your enzymes are going to quit functioning. Chemical reactions are going to quit working, or chemical reactions that don't need to occur will start occurring. And what will happen to homeostasis? It's going to bring your body out of homeostasis. So acidosis and alkalosis, major problem, big deal. Alterations in homeostasis tend to cause alterations in acid-base, or are a major cause of acid-base imbalances for sure. Okay, you guys okay with that so far? So far, so good? Okay, so now we've talked a little bit about acid-base. What I want to do is I want to talk about general types of chemistry real quick. And then we will start on the macromolecules of life. So up to this point, we've just talked about real simple molecules, oxygen molecules, water, and we'll get a little more complicated here. All right. So, place this here. All right. Erase this stuff here real quick. Wish I had more boards. All right. Okay, so there are four major, well, let's talk about types of chemistry first of all. So there are different subdisciplines of chemistry. For the most part, what I've talked about today is something known as general chemistry. General chemistry. All right. This is just the basic foundational knowledge of what atoms are, how they work, what's chemical bonding, et cetera, et cetera. Now, a very important type of chemistry that we need to appreciate involves a specific element, a specific atom. And this atom is unique in that it can bond to four different things at the same time. Okay? So it can act like a backbone, okay? Whatever this atom is, atom X can act like a backbone. It can bond to itself, and it can create a backbone, and then other things can bond to it as well. And so you can see how this specific atom forms the backbone of very complex molecules. What is this atom that I'm talking about? Carbon. Carbon is the backbone of the chemistry of life. Because carbon has 4 valence electrons, right? It can share 4 electrons. It can bond to 4 different atoms at the same time, including itself. So I can have carbon bond to itself, and then it can bond to hydrogens and hydroxyl groups and other types of chemical groups, and it can make more complex molecules. And you can see that carbon is that backbone. Carbon is so important. There's so many carbon-based compounds that there's a whole study, a whole area of chemistry that revolves around studying carbon. What is that chemistry known as? Organic. Organic chemistry is the study of carbon compounds. Okay? Carbon tends to be the backbone of all the macro or the larger molecules in our body. Okay, the larger molecules in our body, the macromolecules, carbon tends to be the backbone. Now, you also have something called biochemistry, which is pretty important for us because that's specifically the chemistry of biological systems, of which the human body is a very complex biological system. Another form of chemistry is something known as inorganic chemistry. So that's a study of atoms other than carbon, tends to be metals in organic chemistry. Okay? And then we also have like physical chemistry, and this is really the study of the physics involved in chemistry and the complex physics behind what makes atoms bond, a field known as quantum mechanics. It's actually quantum mechanics that we use to understand chemical bonding in great detail. But I won't be talking about that in this course. But anyway, there we go. Okay, so let's just review what the macromolecules of life are. What are the major groups? What are the 4 major groups that form the major macromolecules? So now we're going larger. What are they? They fall in 4 things. What's that? Carbohydrates. Good. Carbohydrates. Carbohydrates. That's one of the 4 major groups. What's next?
Lipids.
Lipids. Good. Lipids. These are the fats. Fatty acids. Good. Proteins. Very, very important. Okay. So carbohydrates, lipids, proteins, and then finally... Say it. Somebody said it. Nucleic acids. Nucleic acids. Okay. These are the 4 major macromolecules of life. Okay, so the more simple molecules can form these larger, more complex molecules. The carbohydrates, let's start here and just talk about what they do in general. Okay. The big role of carbohydrates, these are our major energy source. Okay, our cells burn, quote unquote, our cells burn these carbohydrates. They use these carbohydrates to create energy. Okay? It's a very important role for energy. And what is the high energy molecule that our body uses for fuel? Carbohydrates are turned into this. What is the fuel that runs our cells? No, sugar is a carbohydrate, right? Yeah, energy. Carbohydrates are sugars. Sugars are a form or type of carbohydrate. But these are turned into what? What is the energy currency of our cell? What molecule ultimately what molecule does our cells use, like gasoline?
ATP.
ATP. You definitely need to know this. ATP. That is adenosine triphosphate. Okay, adenosine triphosphate. Okay? ATP is like money in the bank. All right? If I have a bank account with $1,500, okay, I know what I could buy, right? Okay, think of ATP as money for the cells, right? Those cells have money in the bank, they can do stuff. If they don't have ATP, they can't do stuff and they die. Okay, they die. They can't pay the bills. Okay. ATP is the fuel. Okay, it's what our cells burn for fuel, adenosine triphosphate. And these sugars are turned into ATP in our cells. That is one of the major roles for carbohydrates. Lipids. Lipids are important for many different things. One thing that lipids are important for is in the cell wall, which we'll talk about in lab. Okay? The cell wall. Okay? You have lipids, cholesterol as well. Cholesterol falls into the lipid category. Okay, and that's what gives the cell wall its fluidity. Okay? Lipids are also important because if you run out of carbohydrates, your body can actually turn lipids into energy, into ATP if it needs to. It's kind of a messy process. There's some problems associated with it, but it can. Okay, so they can be a reserve. Okay, they can also protect. Right, fat can protect organs. Right, act as kind of a buffer, a protection. Okay, so it's a critical component of the cell walls. Cells can use it for energy if they need to. Can protect. Okay, and certain hormones. Okay, specifically steroid hormones come from lipids as well. Okay, proteins. Proteins are super critical throughout the cells. Okay, proteins act as enzymes that catalyze chemical reactions. Okay, proteins act as pumps. They can pump ions in and out of the cell. Okay, proteins act as receptors. Okay, they can signal cells. They can respond to stimuli. They can signal other areas of the body. Proteins are involved in movement. Okay, motor proteins. They can move stuff around inside of the cells. There's a protein called kinesin, and it literally looks like a really long, tall guy with a little head and then two little feet that attaches to what's called the cytoskeleton, and the ATP hits it. And it literally walks along the inside of the cell, carrying stuff around within the cell. It's called kinesin, it's a little motor protein. Okay, so enzymes, pumps, receptors, movement. Proteins are just critical everywhere. Our ability to clot, okay? So we don't bleed to death. That's due to protein clotting factors, by and large. Okay, so proteins are super, super critical. And then nucleic acids are, you can look at these as the blueprint. Okay, these are the blueprint. These act as the blueprint for our cells. Okay? Nucleic acids tell the cell how to make proteins. The code, the genetic code, okay, the genetic code for how cells make proteins, and remember, proteins are what get stuff done, right? That genetic code is made up of these nucleic acids, right? So the genetic code of your cell, the nucleic acids, the proteins gets things done, okay, enzymes, pumps, receptors in and out of your cell, movement, motion. Okay, lipids form a critical part of the cell wall. Backup energy, they can protect. Okay, and certain steroid hormones. And finally, your carbohydrates are a very important energy source. Sugars are a type of carbohydrate that are used to make adenosine triphosphate. You guys okay with that? Those are the 4 major macromolecules of the cell. So what I want to do now is I want to talk about these macromolecules in just a little more detail, if you will. Okay. So let's start at carbohydrates. Okay. Another name for carbohydrate. Has anyone here taken nutrition? Some of you taking nutrition? Okay, so this will be familiar to you. Another name for a carbohydrate is a saccharide. Okay, is a saccharide. Saccharides. And you can have different types of saccharides. If you have just one, that's what we call a monosaccharide. If you have 2 bonded together, that would be a disaccharide. Okay. You can have polysaccharides as well, which would be multiple individual saccharides or individual carbohydrates bonded together. Now, the major carbohydrate that our body uses for fuel, okay, the major one that our body uses for fuel is glucose. Glucose. Okay, glucose. You guys cool with that? That is the major one that our body uses for fuel. Now, when we eat plants, or we eat something that has carbohydrates in it, typically these carbohydrates are not individual glucose molecules, not monosaccharides, but they tend to be polysaccharides. So what you have to do is you have to break those carbohydrates down into their individual glucose molecules or individual saccharides. Does that make sense? And that's part of digestion. And that's actually, have you guys ever like taken bread in your mouth and you ever chewed on some bread? And what happens if you keep that bread in your mouth and you keep chewing it over a certain amount of time and you don't swallow it? Has anyone ever done that? You notice something really interesting happens. It'll start tasting sweeter and sweeter as you chew on it. Why do you suppose that is? Why does that bread start to taste sweeter and sweeter over time?
Your saliva.
There you go. Amylase. Amylase is an enzyme, right, that helps break carbohydrates down. And so as you're chewing on that, your salivary glands are putting amylase in your saliva. You start breaking that bread down into its simple sugars and glucose. And glucose is very sweet, right? Sugar tastes very sweet. So that bread is that complex, okay? The polysaccharides in that bread are broken down into the monosaccharides, and your body can taste. You can taste those monosaccharides, and it gets sweeter and sweeter. How crazy is that? Yeah, so there you go. All right. Now, what makes a carbohydrate a carbohydrate? Well, it's carbon and hydrogen, right? Carbohydrates are hydrated carbons, so it's carbon, hydrogen, and oxygen. And you have to have a ratio, okay? A hydrogen to oxygen ratio of 2 to 1, okay? Of 2 to 1. That's what makes a carbohydrate a carbohydrate. And I'll go back to glucose. The chemical formula for glucose is C6H12O6. That's something you probably want to know. C6H12O6. So 6 carbons, 12 hydrogens, 6 oxygens. And does this reduce to a 2 to 1 ratio? 2 hydrogens for every oxygen? Yes, okay? So that's what makes it a carbohydrate. Does that make sense? Okay? C6H12O6. There's actually a car here around town. The car is a Cube. Is it a Honda? Is it a Honda that makes a Cube? And they have this on their license plate. And so now you guys all know what the joke is. You're in. Oh, sugar, right? Glucose is sugar. Sugar cube. How cute is that, right? There you go. Okay. That's a carbohydrate. This is actually what a carbohydrate looks like. Okay. It has this ring-like structure here. So you have these 4 carbons. Black is carbon, red is oxygen, white is hydrogen. You guys okay with that? Okay, so glucose has this ring-like structure here. Okay, you have these 4 carbons come together at this oxygen here. Okay, you have a carbon that comes off of here. And then whenever you see an oxygen and a hydrogen bound together, that's called a hydroxyl group. You guys okay with that? And whenever you have oxygen, lots of oxygen and hydrogen combined, is that polar or non-polar? That tends to be polar. So do you think that this is actually glucose here, do you think that this glucose molecule would dissolve in water very easily? Yes, it's very polar because it has lots of hydrogen attached to oxygen, right? So this would be polar and dissolve very easily. You guys cool with that? Now in your book, I think there are some pictures of a line structure, and what you can actually do to this molecule is you can break it open and it'll form a straight line. And that's often how it's depicted in textbooks, is as a straight line. So you either see it drawn as a straight line, or sometimes you'll see it in its ring structure, okay? Both are valid ways of looking at it, but there you go. You guys good with that? That's glucose, okay? Carbohydrate. Okay, let's talk about the next major molecule of life, and that is the fatty acids, or the lipids, okay? What differentiates a lipid from a carbohydrate? You guys have your book? Tell me. Yeah, but what chemically? What makes them? What makes them unique? What's going on there with a fat? What's that?
Less oxygen.
Much less oxygen.
The
What?
ratio is reversed.
Is it reversed? What is the ratio?
1 to 2.
1 to 2.
Carbons for hydrogen.
There you go. Carbon to hydrogen ratio is 1 to 2. So there's 1 carbon for 2 hydrogens. Okay, so there's a lot less oxygen. So lipids, okay, lipids in fact have a 1 to 2 carbon oxygen ratio. Okay, so let me give you an example. So here is a carbohydrate. You guys okay with that? You can see it has lots of oxygen and lots of oxygen in there, right? Lots of red. Here is a fatty acid. What do you notice? What does it have a lot of? A lot of hydrogen, a lot of carbon, and not as much oxygen. Okay, this is what we call a fatty acid. The reason that this is a fatty acid is, do you guys see this end part here? This end part here. This is one of the most common acids in the human body. This is what's known as a carboxyl group. A carboxyl group. Okay. That is a COOH group. Okay, it is a carbon that is double bonded to an oxygen. And do you guys see this double bond here to this oxygen? So carbon double bond to the oxygen, and then an OH group, a hydroxyl group, or this is also known as an alcohol group, hydroxyl or alcohol group, OH. And when this thing goes into water, this hydrogen pops off of that oxygen, all right? And that's what makes it an acid. Does that make sense? Why this is an acid? So we call this a fatty acid. But what do you notice? How polar do you think, if you were to compare this to this, the carbohydrate, which one do you think would be much more polar? The carbohydrate. Much more polar, right? Because you have all of this oxygen and hydrogen, right? You only have one area where you have oxygen and hydrogen here on the fatty acid, right? So this is much less polar than this. And this is what makes fats where they don't dissolve in water as well, okay? because they can have these and this is like one of the shorter carbon chains. These lipids can have 10, 15 carbons, very long carbohydrates. So a significant portion of the molecule will be nonpolar. Will it want to dissolve in water very easily? Not really. This part here a little bit, but the rest of the molecule, no, not really. And molecules that do not want to dissolve in water are what we call hydrophobic. Okay, water-fearing. Fats are hydrophobic. Okay, and molecules that do want to dissolve in water are what we call hydrophilic. Okay. Now, some other terms that you guys need to be aware of, and I'm not going to be able to talk about proteins and nucleic acids today because that's a little more complex conversation. So we'll pick that up, I guess, next week. Huh? Pick that up next week. Okay. So, you guys see this okay here? What was I going to say? Totally lost my train of thought. I'll blame it on you guys.
Hydrophobic.
Yes, I was talking about hydrophobic. I was talking about oh, yes. What you need to know. When I look at a fatty acid, if every carbon in the main chain, this is my main chain here. This carbon here is not part of it, just these three here, or these four. If every carbon has a maximum number of hydrogens bonded to it, that's what we call a saturated fat. A saturated fat is where every carbon in the main chain is saturated with a hydrogen. Okay, that is saturated. You guys cool with that? A saturated fat. Now, are saturated fats, my nutrition gurus, are those typically good or bad fats? Why are they so bad? It's because they can stack up on each other. Saturated fats, if I had, imagine that this is one, another one could stack on that, and another one could stack on that, and they could easily stack up on each other. Okay, that's a problem with saturated fats, and they're good at stacking up inside of our blood vessels, right? And causing something known as atherosclerosis or narrowing, buildup of plaque in the blood vessels, right? Saturated fats. Does that kind of make sense? Okay. In saturated fats, do they tend to be liquid or solid? They tend to be solid because they can stack up on each other. Does that make sense? Now, if I replace one of these hydrogens with a double bond, so I have a carbon double bond like this, do you guys see where I have that double bonded carbon there? See that there? I replaced a hydrogen with a double bond, with another bond. Okay, this is saturated, so this would be an unsaturated fat, right? This is saturated, this would be unsaturated. Now, is this going to be more likely to stack up on itself? No, because see how the shape is kind of broken and it has this weird shape, so it's not going to stack up as easily and as neatly. So do these tend to be liquid or solid? These tend to be more liquid, right? These tend to be more solid, right? And the more unsaturated, right? So this would just be a mono unsaturated, right? One bond, but I could have others, and that would be polyunsaturated, right? And be more and more liquid. And just to make matters worse, when we talk about unsaturated fats, I can have a cis and a trans, right? You guys remember the term cis and trans fats? You guys ever heard of that? A cis, right? That refers to your unsaturated fats. Okay? And what that does is that refers to see these two hydrogens here? And see how one hydrogen is up in this direction and the other hydrogen is down in this direction? They're in opposite directions from each other? Right? What do you suppose? Is that cis or trans? What do you think? Yeah, that's trans, right? They're on opposite ends of the carbon. And then if both if the hydrogen is on the same end, we would call that a cis fatty acid, right? So I'd have cis and trans as well. But just to recap, saturated, right? These tend to be the ones we worry about because they can stack up on each other a lot easier, right? Tend to be more solid. Okay. So this is saturated. This is unsaturated. Okay. I've replaced a hydrogen with a covalent bond. And within the unsaturated, I can have cis or trans. You guys okay with that? Does that kind of make sense? So far, so good. The last couple of minutes, there's just one more thing I want to talk about. And unfortunately, I'll have to save proteins and nucleic acids until next week. But I think I've thrown enough at you guys. Huh? Okay, so the next thing I want to talk about is a concept in chemistry known as an isomer. Okay, known as an isomer. And what an isomer is in general, it is something that has the same number of atoms, but they're arranged a little differently. So, for example, this is just a molecule I made up here, okay? So this is a molecule. I've got two carbons here in the middle. I have an alcohol here, or a hydroxyl. I have hydrogen, hydroxyl, and then a CH3. CH3 is what we call a methyl group, okay? So let's see, that would be C, let's see, 1, 2, 3, 4. You guys good with that?
Yeah.
So I have C4, O, let's see, 1, 2. You guys go with that? C4, O2, H, C, let's see here, 3, 6, 7, 8, 9, 10. You guys okay with that? C4, O2, H10. Okay. But let's say that I had another molecule here. Is this the same chemical formula? C4O2H10? Let's see, I should have two methyl groups, right? 1, 2, 1, 2. 3 hydrogen, 1, 2, two hydrogens. Oh. There we go. How's that? There we go. Now, is this the same C4O2H10? But is it the exact same molecule as this one?
No.
It's a different molecule, okay? That is an example of an isomer where you have the same number of atoms, but the atoms are arranged differently, okay? That's what we call an isomer. This is a constitutional isomer. This would be a constitutional isomer of that. There are even other types of isomers where I have a mirror image. So imagine that this is a molecule here, okay? This is a molecule. This is the mirror image of that molecule. So if this molecule looked at itself in a mirror, it'd see this, right? Are these the same molecule? If they were the exact same molecule, I could put them on top of each other and I wouldn't notice a difference, right? Can I put these guys on top of each other?
No.
Shoot, I can't. They're different, aren't they? You cannot superimpose them. This, okay, mirror image is something known as an enantiomer. An enantiomer. And I liken it to, what is the mirror image of your left hand? Your right hand, right? Can you put your right hand on top of your left hand? No. That is what we call an enantiomer. And many molecules have a left-handed form and a right-handed form. Bless you. And in nature, like carbohydrates, only the right-handed form can be used by the human body, and the left-handed form can't. So some people, instead of saying glucose, what we'll do is we will say the body uses the right-handed, or the D. D is what means right-handedness, and we call that dextrose. Dextrose is the right-handed form of glucose, or D-glucose. Carbohydrates, right-handed, and then when we talk about proteins, it's actually the left-handed form of proteins. All right, get out of here.